Okay, very good evening, my dear friends. Today's our eminent resource person, Dr.P.Manjula Madam Garu. A long back ago, Madam has taken one of the important topic. Now, Madam has come back to the field. So, Madam is going to explain one of the important area in the plant sciences, especially CPGET 2024. Madam, please welcome Dr. P. Manjula Madam Garu, Assistant Professor, Department of Botany, Rbvrr Women's College, Autonomous, Hyderabad. So, Madam, please welcome and share your screen. Start your session. Please welcome.
Okay, thank you, sir.
Madam, please click on slideshow.
Madam, sir, 1 minute, sir.
Okay.
It is internal structure of dicot and monocot stem. Okay, so internal structure of dicot and monocot stem. So first one is a dicot stem. So generally, when we take the internal structure of the dicot stem, so it contains outer epidermis, cortex, and steel regions. Okay, so generally the typical example for this dicot stem is the helianthus. So example for this dicot is helianthus, that is sunflower stem is used as a typical example for dicot stem. So it contains epidermis, cortex, and steel. So epidermis, it is the outermost layer, and it is mainly composed of the parenchymatous cells. So this epidermis, which is made up of parenchymatous cells, and usually this layer is single layered, that is only one layer of cells, one layer of parenchymatous cells, which is made up of which constitute the epidermis. So these are closely packed cells and without intercellular spaces. So outer tangential wall, it is thick, and which is, so generally on the epidermis, so thick walled cuticle is present, which deposited on the outside. So this cutin, which occurs as a separate layer called as the cuticle. So this cuticle, which is made up of cutin. So epidermis of young stem, which contains stomata. So generally young stems contain stomata. So epidermis, it is a single layered and it is made up of parenchymatous cells, and these cells are closely packed. And on the epidermis, that is cutin is deposited. So this layer, it is called as a cuticle. So generally on the epidermis, cuticle is present. So next one. So generally on the epidermis, so multicellular hairs are present. So they are called as the trichomes. Okay, they are present on the epidermis. So in herbs, generally these epidermis remains throughout the life cycle. But in woody plants, so generally these epidermis, which is replaced by the secondary growth. Okay, and the main function of this epidermis is, that is, it mainly gives protection, and the presence of this cuticle, which prevents water loss and stomata, which is present on the stem, which mainly helps in the gases exchange, and trichomes and hairs, which provide the protection. So on the epidermis, so the main function of this epidermis is the protection, and due to presence of this cuticle, which mainly prevents the water loss, and stomata, generally on the young stem, stomata are present, mainly helps in the transpiration, and trichomes and hairs, which provide the protection. So next one is the epidermis, which is followed by the cortex. So tissue inner to the epidermis, it is called as a cortex. So in some plants, cortex is simple and undifferentiated. Okay, so all the cells are made up of same type of tissue. That is, it may be a parenchyma or sclerenchyma or cholenchyma. So otherwise, so in some times, so these cortex, which is differentiated into different layers like hypodermis, outer cortex, inner cortex, and endodermis. So first one is the hypodermis. So that is epidermis, which is followed by the layer which is called as a hypodermis, and the layer of tissues that is just below the epidermis, it is called as the hypodermis, and the cells are cholenchymatous. So here the cells, hypodermis, it is made up of cholenchymatous cells, and they are compactly arranged without intercellular spaces. So generally in xerophytes, so these hypodermis, which is sclerenchymatous, that is the hypodermis which is made up of sclerenchyma. So this point is important. So generally in xerophyte, the hypodermis is sclerenchymatous. So normally it is cholenchyma, but it is sclerenchymatous in xerophyte. So this point is important. And in stem with ridges and furrows, the cholenchyma mainly occur below the ridges. So when the stem is irregular, that is ridges and furrows only. So here in this condition, the mainly cholenchyma cells which are present at the ridges. Okay, so in very young stem, the cholenchyma is poorly developed. Usually the chloroplast is absent in hypodermis because here it is made up of cholenchyma. Okay, and rarely cholenchyma sometimes, which may contain the chloroplast. So in xerophytes, the hypodermis is sclerenchymatous. So this is the xerophytic character. Presence of sclerenchymatous hypodermis, it is the xerophytic character. Next, the main function of this hypodermis is which provides mechanical support, and in plants with secondary thickenings, so these hypodermal cells which gives rise to cambium, so which gives rise to the bark. So next hypodermis, which is followed by the outer cortex. So which is here this tissue which is inner to the hypodermis, and mainly these outer cortex, which is the chlorenchymatous. That is, here the cells are made up of parenchyma and which contains the chloroplast, and they are loosely packed with intercellular spaces. Next, in xerophytes, the outer cortical cells form the palisade-like tissue. And the main function is here this outer cortex, that is the photosynthesis. Next one is the outer cortex, which is followed by the inner cortex. So the tissue inner to the outer cortex, it is the inner cortex, and the cells here are parenchymatous. So inner cortex, which is made up of parenchymatous cells, and they are loosely packed with intercellular spaces. And the main function of this inner cortex is storage of carbohydrates. So the main difference between this hypodermis in outer cortex and inner cortex. So hypodermis mainly it is made up of cholenchyma and it gives mechanical support, whereas the outer cortex, which is made up of chlorenchyma, that is they contain the chloroplast and mainly helps in the photosynthesis, whereas the inner cortex, which main function is storage of the carbohydrates. So next one is in hydrophytes, the cortex is with plenty of air cavities, that is called as the aerenchyma. Okay, so this presence of aerenchyma, it is a characteristic feature of the hydrophytes. So aerenchyma mainly helps in gases exchange and which provides buoyancy to the plants. So sclerenchymatous patches which occur on the cortex of eucalyptus, eugenia, and ficus. So generally the patches in the cortex. So in general, they are seen in the eucalyptus and eugenia and ficus, and the secretory cavities which are also present in the cortex of eucalyptus, and resin canals which occurs in the cortex of the anacardium. So here these are some special characters. So here the cavities which are present in the cortex. So latispherous cells which occur in the cortex of the latex producing plant. So these all comes under the again the secretory tissue. So latispherous cells, resin canal. So these are all present in the cortex. So the main function of the cortex is which provides mechanical support, and during secondary growth, this hypodermal cells which gives rise to cork cambium from the bark, and bark formation mainly helps in the formation of gives rise to cork cambium, so which produce the bark. So chlorenchymatous cells in the outer cortex can do photosynthesis, and parenchymatous cells of the inner cortex which mainly helps in the storage of the carbohydrates. And cortical cells also stores ergastic substances. So the cells of the cortex, they are called as the cortical cells, and they store the ergastic substances. So resin canals, latex canals also occur in the cortex. Next one is the first hypodermis, which is followed by the outer cortex. Outer cortex is followed by inner cortex, and inner cortex which is followed by a single layered the endodermis. So innermost layer of the cortex, it is called as the endodermis. So which is very distinct in pteridophytes, but it is not distinct in gymnosperms and angiosperms. So endodermal cells which accumulate the starch as the starch grains, hence it is also known as the starch sheath. So it is single layered parenchyma, which is made up of parenchymatous cells, and which stores the starch. Hence, it is called as a starch sheath. So, starch sheath is nothing but the endodermis. So, generally, endodermis, it is called as the starch sheath. So, if distinct, endodermis is uniseriate with barrel-shaped cells. So, when we observe this endodermis, it is uniseriate, single-layered with barrel-shaped cells. So, cells of the parenchymatous and compactly arranged. So, here it is made up of single-layered parenchymatous cells, and they store the starch. Next one is, so have the characteristic thickenings in the radial and inner tangential walls. So these thickenings, they are called as Casparian thickenings. So they are also known as the Casparian band or layer, Casparian layer. So this band which is composed of suberin and lignin. So both are impervious to water. They cannot absorb the water. So block the passage of water and solutes through the protoplast and endodermal cells. So sometimes here the Casparian thickenings or bands are present in this endodermis. So they do not allow the water and other solutes to the inside. So the main functions of the endodermis are, exact function of the endodermis is not known, but they do not allow the passage of water and can store food materials, that is as a starch grain. So next one is the stele, that is vascular bundles region. So stele, it is the central vascular cylinder and mainly composed of 4 components, that is the pericycle, vascular bundles, medullary rays, and pith. So pericycle, so endodermis which is followed by the pericycle, it is the outermost layer of the stele and which is located next to the endodermis, inner to the endodermis, and show variation. So in some plants, this pericycle is absent, but usually multi-layered. So here the pericycle which is made up of multi-layered, and that is 3 or more layered cells, and pericycle may be completely parenchymatous or completely sclerenchymatous or mixture of parenchyma and sclerenchyma. So here the variation in the pericycle may be seen. So generally pericycle, it may be made up of totally parenchyma, sometimes sclerenchyma, or sometimes both a mixture of this parenchyma and sclerenchyma, that is alternate. Only in the vascular bundles region sclerenchyma is present, and in other region parenchyma. Next one is so here vascular bundle. Next one is a vascular bundle. So it is called as a fascicle. Vascular, another name for this vascular bundle is the fascicle, and which is located inner to the pericycle and which is developed from the procambium. Okay, vascular bundle which is made up developed from the procambium, and number of vascular bundles is limited. That is 6 to 8 vascular bundles are present in the stem. Dicot stem. So arranged as a broken ring. So it is looks like a arranged as a broken ring. And these vascular bundles mainly composed of xylem and phloem. So xylem which is placed inner to the cambium and phloem is placed outer to the cambium. So vascular bundle which contains cambium. So inside to the cambium, that is xylem is present, and so this one is the okay, so this one is the vascular bundle. So here this one is the cambium. So outside this cambium, phloem is present, and inside xylem is present. So they are arranged. So here the number of vascular bundles, that is here 5 and 3, 4, 5. So 10 vascular, so minimum, that is 6 to 8 vascular bundles are present. So here the vascular bundle, the position of these vascular bundles, that is the conjoint, that is xylem and phloem is present on the same radius, and open, that is cambium, presence of cambium, that is in between the xylem and phloem. If the cambium is present, so it is called as the open. And xylem is endark. Endark means that is protoxylem towards the inner side and metaxylem towards the outer side. And collateral are bicollateral. So here the bicollateral means here. Collateral means usually one patch of xylem and one patch of phloem. It is called as a collateral. That is on the cambium, one side xylem is present and another side phloem is present. Bicollateral means here the xylem layering in between the two phloem patches. A median patch of xylem on either side of this xylem, phloem is present. That is bicollateral. And bicollateral vascular bundle. So this is a special character which is seen in the cucurbitaceae members. Okay, bicollateral vascular bundles. So next one is the xylem. So the main function of xylem is conduction of water and minerals, and mainly this xylem which contains, which is made up of tracheids, vessels, fibers, and parenchyma. So that is again this xylem is differentiated into protoxylem and metaxylem. So first formed xylem, it is called as a protoxylem, and which is arranged towards the center. There are very less amount of tracheids elements are present in the protoxylem, and large amount of parenchyma is present. And here the tracheids elements with narrow lumen. So here the tracheids elements are present. So they with narrow lumen, and annular or spiral thickenings are seen on the tracheids elements. And next one is the metaxylem. So later formed after the protoxylem, and here more tracheids elements than the protoxylem, and cell with large lumen. So the difference between this proto and meta. So here the large lumen is seen, here the narrow lumen. Okay, so cells with reticulate or pitted thickenings are seen in the metaxylem, whereas the annular or spiral thickenings are seen in the protoxylem. So these differences are also important for the in view of competitive exams. Annular or spiral thickenings generally they are seen in the protoxylem, whereas the reticulate or pitted thickenings they are seen in the metaxylem, and arranged towards the exterior, that is generally metaxylem towards the outside. So the main function of the xylem is conduction of water, conduction of minerals, and which provides mechanical support due to presence of these xylem fibers, and xylem parenchyma mainly helps in the storage of food materials. Next one is the phloem. So the food conducting tissue of the vascular bundle and composed of sieve tubes, companion cells, parenchyma, and fibers. So here also differentiate into protophlloem and metaphlloem. So protophlloem means first formed and arranged towards the periphery, and metaphloem which is differentiated after the protophlloem and which is located near to the cambium. So cambium. So this point is important. So if the cambium is present, then only here the secondary growth will takes place. So due to so generally in Dicot stems, the secondary growth will takes place. So due to presence of this cambium. So it is a layer of meristematic tissues which present between the xylem and phloem. So this cambium, the location of this cambium is which is present in between the xylem and phloem. And this cambium, which is a meristematic tissue. Meristematic tissue means it undergoes continuous state of division. Okay, so it is also known as the vascular cambium or vascular cambium. So cambium also known as the vascular or vascular cambium. So it is the remnant of the original procambium remaining. So cells are parenchymatous and thin-walled. So the cambium cells, they are parenchymatous and thin-walled, and cells with dense cytoplasm and prominent nucleus. So generally secondary growth in dicots occurred due to cambium. So here the secondary growth which takes place in the dicot stem, it is due to the presence of cambium. So vascular bundles with cambium, it is called as the open vascular bundle. So open vascular bundles means cambium is present. So next one is the steel, steel, that is medullary rays. Okay, so vascular bundles are xylem, phloem. Next one is the medullary rays. So it is also known as the pith ray. So occurs between the vascular bundles. So the main function, which is composed of parenchymatous cells, medullary rays, they are made up of parenchymatous cells, and the cells are radially elongated. So these are the medullary rays. So during the secondary growth, the cells of the medullary rays gives rise to intervascular cambium. Okay, intervascular cambium, which is developed from this medullary ray. And the main function is which allows radial transport of water and provide intervascular cambium during the secondary growth. So during the secondary growth, that is vascular and intervascular cambium forms a ring-like structure and which produces the secondary xylem towards inner side and secondary phloem towards the outer side. So next one is the pith or medulla. So it is also called as a medulla. So which is located towards the inner side of the vascular bundle, and here exact central portion of the stem. So this central one, it is the pith and mostly parenchymatous. So this medulla, which is made up of parenchymatous cells, and it may be loosely arranged. So sometimes these wall thickenings are occurs, and the cell of the outer region smaller and inner region larger. So the cells here, the variation may be seen. The outer region smaller, the cells are smaller in size, and inner cells, they are larger in size. So it is made up of parenchyma. So in some plants, the pith is replaced by the large air cavity called as a pith cavity. So the function of the pith is that is the storage of food materials, because here it is made up of parenchymatous cells, and it main helps in the storage of food materials. So this is about the Dicot stem. So generally in Dicot stem, that is the epidermis. Epidermis is covered with thick cuticle, and here the trichomes are present. Next one is the cortex. Here the cortex is differentiated into hypodermis, general cortex, or that is hypodermis, and outer and inner cortex. Next one is the endodermis. So next endodermis, which is followed by the pericycle. Okay, so endodermis is single layered, and it is also known as the starch sheath. Next one is the pericycle. So it is multi-layered with the sclerenchymatous cells are intermediate parenchymatous cells. So here the variation may be seen. So next one is pericycle, which is followed by the vascular bundle. So generally in dicots, there is the vascular bundles are conjoint, collateral, and open. Okay, so next one is the cambium is present. So next one is it may due to presence of these cambium, which mainly helps in the secondary growth, and pith also present with the main function is storage of the food materials. So next one is the internal structure of monocot stem.